Hi class, happy snow day. We're going to go through active transport and bulk transport in the lesson today, just so that it gives us a little bit more time tomorrow to ask questions and start studying for not start studying, but use class time to study for the unit test. So the other day we talked about passive transport. So when we're talking passive transport, remember we're going from an area of high concentration to an area of low concentration without energy. Where active, you're moving from an area that's already has a high concentration from an area of low concentration, which molecules don't want to go because you're going to kind of be confined with space. So what's needed is you need this ATP, which is going to be our energy molecule. It's the idea where we talked about students in the classroom, where you had 200 kids in our classroom and there's like 2 or 3 in the hall. They're not really going to want to pack in that classroom, so they need the push. The push is energy in form of the cell. It's going to be your ATP. So just remember, active transport, always going from an area of low concentration to high concentration using energy, which is going to be in the form of adenosine triphosphate or ATP molecule. That is like our energy carrier. What happens is this ATP molecule undergoes a hydrolysis reaction. So you're adding in that water, and then you break apart that last phosphate group, and it becomes ADP now, adenosine diphosphate. And when you do that, you actually release energy. And so this reaction releases energy. How this works with our active transport is we're going to look at what's called the sodium-potassium pump. It's the most kind of studied, well-known primary active transport. It's what our nervous system uses to allow for the neurons to send signals down. So this is going to be our primary active transport. So what's happening? If you look at it, you can see it has 3 sodium ions moving from an area of low to high, which it doesn't necessarily want to do, so it's going to need the push. ATP undergoes a hydrolysis reaction. The phosphate is left attached to that carrier protein, and in doing so, it causes it to change shape, and then it releases those 3 sodium ions to the outside of the cell. Okay? Then the phosphate gets being released at the same time you have 2 potassium ions moving in. Once that phosphate's released, the carrier protein changes its shape, and then you now have brought 2 potassium ions into the cell. So simultaneously, we're bringing 3 sodium ions outside the cell and 2 potassium ions into the cell by using ATP in a hydrolysis reaction to change the shape of your carrier protein. Sorry. Okay, so let's kind of go through it one more time in this picture. Okay, so you can see here, you have your 3 sodium ions going in. ATP undergoes a hydrolysis reaction, becomes ADP, the phosphate's attached, changes the shape of your carrier protein. These sodiums are released outside of the cell. And it has now moved those sodium ions from an area of low concentration to an area of high concentration because of the energy. Then 2 potassium ions are going to move in. Your phosphate's released, changes back the shape to where it was. Sorry, my hand hit the side. There we go. Okay, changes the shape. 2 potassiums released to the inside of the cell. And so it works together to allow us to move 3 to 2 for your ions using your energy. There are some protein structures and molecules that need the primary active to function in order to allow for it to function. So this glucose co-transporter is what's called as secondary active, in that it relies on your primary active in order to move glucose. So if we're looking, you can see we're going to talk primarily about glucose for this slide. You're going from an area of low concentration to an area of high concentration. And when you look at it, This transport protein is not using energy. So how is it able to move from an area of low concentration to high concentration without energy? It actually moves based on what's called an electrochemical gradient. So it's now we're not just having a high concentration versus a low concentration, we're having a high difference in charges. So it's not just concentration, it's concentration and charges. Because we know that your sodium ion carries a positive charge. So now we're moving, what happens? So you have your primary active happens, 3 sodium ions move outside of the cell, your 2 potassium move in, but that doesn't really affect the secondary active. When these sodium ions, they want to move back because they want to go from their area of high concentration to their area of low. Because it's very charged and really concentrated, glucose, which is also polar, wants to buddy up with it. And so those sodiums move through and they pull glucose through at the exact same time. So glucose doesn't necessarily use the energy directly, but it cannot move through the secondary active pump without the energy being used in the primary active pump. So they kind of coincide together. They need each other to function. So when you're looking, you have to have that electrochemical gradient. So you're getting the concentration plus the charge that allows for it to pull that glucose molecule together. Okay, so again, we're just kind of showing you the same secondary active transport with that glucose that we showed you. This is just another example. So this one is using hydrogen and sucrose. So it's the same idea where now you use your energy with your primary active, and then the electrochemical gradient is going to be pulling it through for your secondary. Okay, so you have similar pumps. You always have to have your primary functioning or else the secondary doesn't function because you need that electrochemical gradient being created from the active transport. Once you have that buildup of not just charges and concentration, you need them together, it can pull across the other ones. And so then just as we remember and kind of review, remember passive is always going from an area of high concentration to low concentration without energy. Where you're active always needs energy because it's going from low to high. And you always kind of are trying, except for the form of active, to equal out your concentration gradient, especially when you're talking about your passive transport. That's why it's going from an area of high to low. The last few types of transport are called bulk membrane transport. They are going to use the cell membrane to make vesicles and to move substances in and out of your cell. And so endocytosis makes that you are bringing stuff into the cell. And I will kind of show you in a picture on the next slide. Exocytosis is moving stuff out of the cell. Makes sense. Enter, exit. Kind of works with it. And what it does is it creates little vesicles. And so if you kind of look at this picture here, you can see, we'll do endo. They're kind of flipped from what you guys have on the slide before. So you can see this is endo. So you're bringing materials into the cell. Okay, so you're having a larger substance that can't just pass through your phospholipid membrane. So what happens is that substance causes the membrane to pinch off, and so it kind of folds in on itself and it will eventually pinch off into a vesicle. That's bringing larger substance into the cell. And what it will do is this side will cause the cell membrane to shrink a little bit. But when you do exocytosis, it causes the cell membrane to go a little bit bigger. So if they don't happen simultaneously, you're either going to shrink or expand the cell too much. So endo and exo constantly have to be happening together. And so exocytosis is where you have stuff leaving the cell or exiting the cell. So you can see here you have a vesicle happening at usually your endoplasmic reticulum Golgi apparatus, and it creates a vesicle that's going to fuse with your phospholipid membrane to allow for these substances to be released outside of the cell. So kind of just similar to what we just talked about, when you're looking at endo, remember that means it's going to enter the cell. You can see the larger substance, you can have larger molecules, you can have dissolved substances depending what it is. It goes to the cell membrane. The cell membrane then is going to create a vesicle and bring it into the cell to be used and distributed where it needs to. There's two types of endocytosis. Phagocytosis, which is cell eating. So it's engulfing larger particles. Sorry, I thought I hit the highlighter. So the larger particles, where pinocytosis is going to be cell drinking. You're looking at your fluids and your liquids. They both are pinching off of the cell membrane to create a vesicle. It's just talking about which substances are in it. The last form of endocytosis is receptor mediated. What that means is you're still going to have the membrane pinching off to create a vesicle, but now you have proteins on the outside of that cell membrane that substances need to bind with in order to trigger the folding in of the membrane to create that vesicle. So you need the receptor to bind with its target, then you're going to get that vesicle forming inwards. They all reuse energy one way or another. We're just not going to get into the little details about it. Just knowing the overall for endo and exo is all we kind of need for this course. So exo is going to be the opposite of your endocytosis. So this is where you're getting materials leaving the cell. So you're going to exit the cell. Okay, and so the idea is that you have your vesicle coming up. It's going to fuse with your cell membrane to release those substances out of the cell. It could be hormones, neurotransmitter, enzymes, just depends what we want. But remember, if we don't have both happening together, you're going to end up with a change in your cell membrane. So step by step for your exocytosis, you can see here from the endoplasmic reticulum, you're going to your Golgi, then you're going to have your vesicle, which then fuses with your membrane to cause exocytosis happening. Okay, so that's kind of the rest of the biochem unit. You are able to kind of finish off study. Email me if you have any questions. I kind of posted the textbook questions and stuff for you here to make sure that you kind of know what you're doing. We will go over any questions you guys have tomorrow. Thank you.